experiment, we are going to study the process of nitrogen fixation in leguminous plants. The material required for the purpose are root nodules of leguminous plants, a petri dish, a brush, water, glass slide, and a microscope to observe the slides that you have made. So for the root nodules of leguminous plants, you may just pull out a bean plant that you might have grown yourself or you might have it in your garden, and you will find structure like the one I'll show you in the diagram, small round little balls present on it. So the purpose of nitrogen fixation is to convert the atmospheric nitrogen into the nitrates or nitrogenous compounds in the soil, which can be utilized or which can be absorbed by the plants to build up proteins. So basically, there are certain microorganisms living in the soil, like the symbiotic bacteria, which are the nitrogen fixing bacteria, and certain blue-green algae, which have this special ability of converting the nitrogen in the atmosphere to these compounds in the soil. In addition to this, whenever there is thunder and lightning, that also helps in converting the atmospheric nitrogen into nitrates, which are then washed down to the soil with the help of rain. Leguminous plants include plants like pea, soybean, and all these plants, all the pulses that you know, they are all considered to be leguminous plants, and they have in their root nodules rhizobium bacteria, which is a nitrogen fixing bacteria, which has this special ability to convert atmospheric nitrogen into nitrogenous compounds in the soil. So growing leguminous plants is a very good method of replenishing the fertility of the soil in a natural way, and that is why farmers, they generally cultivate leguminous crops after wheat, so that the fertility of the soil can be restored in a natural way without adding many fertilizers. So this is what the root nodules look like. You will find these round ball-like structures on the roots of any pulse, like if you pull it out from the soil, and these are what are known as the root nodules. Now you can prepare a slide of these root nodules by crushing them in some water, and then using a brush to pick up that smear from the petri dish and place it on a slide, cover it with a cover slip, and then observe it under a microscope. So you may observe three different kinds of organisms here. If you observe something like this, I'm talking about the first figure. Let's call this figure A. This is a microscopic image of rhizobium. So rhizobium would look something like what you're seeing in A. This is the microscopic image of Azotobacter. So Azotobacter is a free-living bacteria which has the ability of converting atmospheric nitrogen to nitrates in the soil. And you can also observe something like this, figure C, which is that of Clostridium, which is again a free-living bacterium present in the soil. So these are three different images that you would be observing under a microscope of these nitrogen fixing bacteria.